Hello, everybody, and thank you for joining us for the webinar today. My name is Wayne Kotkamp. I'm the Circular Dichroism Product Manager for JASCO USA. I'm very pleased to be introducing Dr. Joseph Cardos today, who will give the presentation. Professor Cardos has been leading the Neuroimmunology Research Group at the Department of Biochemistry at ELTE Eötvös Loránd University in Budapest. His primary research includes protein folding and misfolding, the structural and thermodynamic principles behind protein aggregation and amyloid formation, proteomic studies on the role of protein aggregation in neurodegeneration, immune complement system activation, and the complement system's function in the brain. He's developed innovative methods to investigate the thermodynamics of amyloid formation and to analyze protein spectroscopy structure and fold using circular dichroism spectroscopy. Together with his colleague, Dr. Andreas Miskanai, he's developed the BeStSel method, which allows for the estimation of protein structure from circular dichroism spectra with superior accuracy. Their publications on the method have been cited in over 2,000 scientific papers and become integral to the curricula of numerous prestigious universities. Today's presentation was recorded in advance, and when completed, we'll be hosting a live question and answer period with Dr. Cardos. If you have questions, please enter them as soon as possible during the presentation using the Q&A icon at the top of the screen. We'll now begin the presentation.
Welcome. I'm József Kardos from Eötvös Loránd University, Budapest, Hungary. Today I will discuss the structural analysis of proteins using CD spectroscopy. First, I will introduce the role of low-resolution structural analysis, including the investigation of secondary structure composition. Then I will explain the basics of CD spectroscopy and its application to protein molecules. I will address challenges in secondary structure determination, introduce the BeStSel method in detail, and highlight the advantages and applications of CD spectroscopy. Finally, I will present case studies demonstrating how CD combined with BeStSel enhances structural analysis. For those unfamiliar with CD, we provide a brief introduction. Light, as electromagnetic wave, can be plane polarized, with its electric field oscillating along a fixed axis. It can also be circularly polarized, where the electric field vector rotates around with a constant amplitude. Right circularly polarized light rotates clockwise, while left circularly polarized light rotates counterclockwise. This animation, created by András Szilágyi 30 years ago, illustrates the concept effectively. When right and left circularly polarized light waves have equal amplitudes, their superposition results in plane polarized light due to cancellation of the horizontal components. However, when the amplitude of the two circularly polarized lights are different, the horizontal components do not cancel out, creating elliptically polarized light. This effect occurs when the sample absorbs left and right circularly polarized light differently. The phenomenon is called circular dichroism. Based on these, circular dichroism can be expressed as the absorption difference between the left and right circularly polarized lights, or with the ellipticity of their superposition. The absorption difference normalized with the molar concentration and the path length in centimeter will give the extinction coefficient difference, which is the delta epsilon, in per mole per centimeter unit. The ellipticity, which is the ratio of the small and large axis of the ellipse, equals to tangent theta. This theta angle is in the range of millidegrees, and this is used to describe the ellipticity in CD as an alternative of the absorption difference. The normalized ellipticity will be the molar ellipticity, which historically is given in degree centimeter to the square per decimal. Using these units, there is a factor of 3298 between delta epsilon and the molar ellipticity. In the case of proteins, the delta epsilon and the molar ellipticity is normalized for the number of residues, more accurately, the number of peptide bonds. In this case, molar ellipticity referred to as mean residue ellipticity. Chiral molecules or chromophores in asymmetrical environment can show the phenomenon of circular dichroism. Key biological macromolecules with CD spectra include nucleic acids and proteins. This slide illustrates their structural diversity. Our focus here is on proteins. Before going into this, we switch to the lab where our colleague will execute a CD measurement on a protein sample to demonstrate how difficult a CD measurement is.
Welcome everyone. We are going to collect the CD spectrum of a protein on this instrument, which is ready to use. The lamp is warmed up. First we will measure the baseline and then the protein sample. We fill the cell. This is a typical measurement in a 1 mm cell. The protein concentration will be 0.1 mg per milliliter. We put the cap on, place the cuvette into the cell holder, and we have to set up the parameters for the measurement. We will measure in the range of 260 nanometer down to 190 nanometers with 0.1 nanometer data pitch. We will use here a continuous scanning mode with 50 nanometer per minute scanning rate and with 2 seconds data integration time. We use 1 nanometer bandwidth and we're gonna accumulate 10 scans to reduce the noise. So we OK the parameters. We will collect 3 channels: the CD signal, the photomultiplier voltage, and the absorbance. We started the measurements and it will take about 15 minutes at most to collect the baseline, and then we start the protein sample measurement afterwards.
Thank you. Safety is important, but perhaps the helmet is not necessary. While the measurement is running, we continue our discussion. In the near UV range, the CD signal primarily originates from the aromatic side chains, mainly tryptophan and tyrosine residues. Their electronic transitions are sensitive to their microenvironment, making near UV CD useful for detecting changes in the protein's tertiary structure. However, interpreting structural details is challenging, and spectra are often used as fingerprints of conformational states. Near UV CD can also indicate structural disorder, as solvent-exposed aromatic residues in disordered proteins tend to have near zero signal. On the right, we compare a wild-type enzyme at the bottom to its point mutant, which is shown at the top, showing that the wild-type molecule retains its structure under acidic conditions, whereas the mutant is highly sensitive to pH changes. In the far UV region, mainly the electronic transitions of the peptide bonds contribute to the CD spectrum. These are the sigma-sigma star at very low wavelengths, and the pi-pi star and n-pi star transitions in the less far UV regions. At the bottom, CD spectra of proteins with characteristic more or less pure secondary structure types are shown. Red represents a protein with high alpha helix content, blue is a parallel beta sheet, green is antiparallel beta, purple is mostly disordered, and orange is for a protein with polyproline helix. Obviously, the CD spectra are highly dependent on the secondary structures and thus might be usable to estimate the secondary structure composition. We assume that in the case of a mixed structure, the different secondary structures contribute to the overall spectrum with their characteristic basis spectrum in the ratio of their fraction in the structure. This way, the overall spectrum is the linear combination of the basis spectra. This slide illustrates the structural organization of polypeptide chains. Amino acids are linked by the peptide bonds, and depending on the backbone hydrogen bonding pattern, different secondary structures are formed. The most common include alpha helices, parallel and antiparallel beta sheets, and turns, though other secondary structures also exist with lower frequency. Seeing the large variety of the CD spectra of proteins with different secondary structure composition, it is feasible to use the CD data to determine protein secondary structure composition, provided that a suitable analysis algorithm is available. At the top, we present CD spectra from various proteins with approximately 50% alpha helix content, demonstrated the uniformity of alpha helical spectra. At the bottom, we show spectra from proteins with around 50% beta sheet content, but no alpha helix, which exhibit significant spectral diversity. This suggests that beta sheet contributions to CD are highly variable, complicating secondary structure estimation. The underlying reason is the structural diversity of beta sheets, which can vary in strand number, length, parallel or antiparallel orientation, and degree of twist. Historically, this variability was considered a fundamental limitation of CD spectroscopy, leading to a perception that CD was reliable for alpha helices but inaccurate for beta sheets. Over recent decades, multiple algorithms have been developed to estimate the secondary structure from the CD spectra. However, beta sheet estimation remains particularly challenging. To illustrate this, we tested existing algorithms on an amyloid fibril sample composed predominantly of parallel beta sheets, with its high-resolution structure determined by solid-state NMR, containing 50% parallel beta sheet. Unfortunately, none of the algorithms predicted accurately this structure from the CD spectrum, often erroneously assigning high alpha helix content. We systematically investigated this issue using a reference dataset of proteins with high-quality CD spectra and known atomic resolution structures. Our focus was on the impact of beta sheets' parallel or antiparallel orientation and their twists. The top left panel shows the distribution of beta sheet twisting angles across antiparallel beta structures, while the top right panel presents the distribution for parallel beta sheets. Below, individual examples illustrate variations in twist. Lectin has relatively planar beta sheets, whereas trypsin inhibitor A features highly right-twisted sheets. Their CD spectra, shown on panel D, are strikingly different, almost inverse, highlighting the strong influence of twist on CD signals. Panel E displays calculated basis spectra for antiparallel beta sheets with different degrees of twist, revealing a continuous spectral distribution between the left and right twisted sheets. Parallel beta sheets exhibit a narrower, more uniform twist distribution, but reference data for these structures remain limited. Based on these observations, we developed an improved algorithm that distinguishes between parallel and antiparallel beta sheets and further classifies antiparallel sheets into 3 categories based on their twist. Our method is built on the SP175 reference set from the protein CD database, supplemented with additional beta sheet-rich proteins for which high-resolution structures are available. We computed secondary structure compositions and beta sheet twists for these proteins. Parallel and antiparallel beta sheets are separated, and the antiparallel ones are divided into 3 twist categories. Altogether, we have 8 components, as will be shown on the next slide. By least square method, optimized sets of basis spectra were calculated on the reference spectra set. During optimization, a subset of reference spectra is selected that provides the best estimation on the entire reference set for a certain secondary structure. The final basis spectra are used to determine the secondary structure of any unknown protein by linear combination. Here we present the 8 secondary structure components used in BeStSel and their relationship to DSSP classifications. DSSP is commonly used to annotate secondary structures in high-resolution structures, but its components are not suitable to use directly for CD spectroscopy. There are components with low presence and negligible spectral contribution. These minor categories are grouped into the Others category in BeStSel, which mainly represents disordered structures. Notably, in BeStSel, antiparallel beta is divided into 3 groups: left-twisted, relaxed or slightly right-hand twisted, and highly right-twisted, and the parallel beta sheet is a separate component. Additionally, alpha helix is classified into 2 groups. Helix 1 is the middle, regular part of the helix, and helix 2 accounts for the ends of the helices, where the hydrogen bonds are not satisfied up and downwards along the backbone and have different spectral contribution. The turn component is identical to that of the SSP. On the bottom, optimized basis spectra are shown. At the top, we show a cross-validated analysis of BeStSel's accuracy across different wavelength ranges using the SP175 dataset. To avoid showing a large table, BeStSel is compared to the three most frequently used algorithms, CELCON, CONTIN, and CDSSTR. The RMSD values indicate the accuracy of secondary structure fraction estimations for different structural components. To make the comparison possible, the BeStSel components are added up to helix, beta, and turn plus other components. We can conclude that, besides beta sheets, BeStSel overperforms the other algorithms for any secondary structure types, including the alpha helix. At the bottom, a comparison is shown on an independent protein set containing difficult to estimate, mostly beta-structured proteins. In the comparison, the best performance for the given secondary structure type among the previously available algorithms are shown. While the previous algorithms struggle, BeStSel maintains similar accuracy to that observed on the reference dataset, confirming its robustness. Using BeStSel, the secondary structure composition of any proteins can be determined with improved accuracy, and it can deal with the diversity of the beta sheets. CD spectroscopy, when paired with a suitable analysis method, offers several advantages as an experimental technique. It provides structural information in solution, accommodates a broad range of protein concentrations, it is rapid, straightforward to analyze, and once an instrument is available, it is cost-effective compared to alternative structural methods. CD spectroscopy can be used to verify the proper folding of recombinant proteins, perform quality control measurements before time-consuming and costly structural techniques, follow the effect of the environmental conditions on the structure, investigate the stability of the proteins, follow protein folding, and study conformational changes associated with protein interactions. Now, with the proper analysis, we can use the technique to study protein aggregation, amyloid fibrils, membrane proteins, and IDPs. CD is widely used in biotechnology, molecular biology, in the pharmaceutical industry, and other fields for protein characterization. We present here some first examples on the use of BeStSel for secondary structure estimation for beta sheets. A great example is the amyloid beta peptide fibrils associated with Alzheimer's disease. While other algorithms fail to analyze this type of structure, BeStSel provides an accurate structural determination. The comparison with the solid-state NMR structure is shown here, and at the bottom you can see how other algorithms struggle to fit the fibril spectrum. Polyglutamine, which is linked to Huntington's disease, can also form amyloid fibrils. By using CD analyzed with BeStSel, we can study how the length of the polyglutamine repeat affects the fibril structure. As shown at the top, long repeats form antiparallel beta sheets, as the extended glutamine sequence allows intramolecular beta sheet formation. In contrast, short polyglutamine repeats result in parallel beta sheet structures. Professor David Eisenberg's group determined the X-ray structure of microcrystals of the GNNQQNY peptide derived from a yeast prion protein. In the crystal structure, the peptide forms parallel beta sheets, leading to the assumption that fibrils in solution adopt the same structure. However, CD measurements reveal a different story. The fibrils actually form an antiparallel beta sheet structure, demonstrating the importance of experimental validation. Probably the baseline has been already measured in the lab. Let's see what's the situation there.
Hi, I can tell you that we have just finished the baseline measurement, rinsed the cell, filled the next sample, the protein sample, and we start the measurement with the same parameters, and in 15 minutes all the data will be ready for analysis.
Okay, thank you. Let's continue the presentation. One major advantage of BeStSel is its ability to distinguish 8 different secondary structure components, which provides enough information to predict protein folds. Since a protein's fold is defined by the specific arrangement of secondary structure elements forming its tertiary structure, BeStSel opens up new possibilities for structural classification. For fold prediction, we use the CAT protein fold classification system, which organizes protein structures hierarchically into 4 levels: class, architecture, topology, and homology. The protein data bank PDB structures can be categorized according to this system. Here we can see the 4 classification levels and subprotein examples for decoration. The concept behind fold prediction is visualized here in 3 dimensions. BeStSel results can be compared to known protein structures in the PDB based on their secondary structure composition. In this protein fold space, represented by secondary structure components as axes, each protein is represented as a single point. By identifying the closest points or those points that are within the expected error of the BeStSel secondary structure estimation by their Euclidean distance, we can find the most similar structures in the PDB. By looking up the fold classification of those structures in the PDB, we can predict the protein fold of the unknown protein. Thus, the fold can be predicted from a single CD measurement as an experimental technique, which might have advantages over the purely bioinformatics tools, as it accounts for the environmental conditions, mutations, protein stability issues, etc. Here are 3 examples of proteins with known 3D structures, demonstrating how fold prediction can be used to build a homology model based on the CD results. First, the CD spectra were analyzed using BeStSel to determine the secondary structure contents. Then the fold prediction was performed, and corresponding 3D structures were selected from the PDB to best match the BeStSel results. The target proteins are shown in green, while the chosen homologues, which can be used for homology modeling, are displayed in gray. The strength of this approach lies in its reliance on experimental data rather than purely computational predictions. Experimental verification of protein fold predictions is crucial. While rare, there are some examples where 2 homologous sequences adopt completely different folds. The molecules at the top are examples of this. Despite a 62% sequence identity, the folds are entirely different. At the bottom, we can see a game with mutations. A single point mutation can convert alpha helices into beta sheets, another mutation can revert it back, and yet another can once again induce beta sheet formation. In these studies, CD spectroscopy was employed to confirm the changes in secondary structure composition. We created a web server which will have the 10th anniversary this year for the secondary structure analysis of CD spectra using the BeStSel method. The server includes various modules to assist users in analyzing their CD spectra and compare their results to the structure of other molecules. The most important blocks are the single and multiple spectrum analysis. There is a module for fold recognition and another to get the extinction coefficients for protein sequences to determine the protein concentrations directly from the CD measurements from the absorbance at 205 or 214 nanometers. Additionally, users can compute BeStSel secondary structure components for PDB structures, facilitating comparison with experimental results or high-resolution models. These models can come from molecular dynamics simulations or AlphaFold predictions. CD spectroscopy combined with BeStSel provides a straightforward experimental method to validate high-resolution models. In the use of CD for verification of 3D models, 2 approaches can be used. What is straightforward, to our opinion, is that we measure the CD spectrum of the protein, analyze it for its secondary structure composition, and compare the results to the structures from molecular dynamics simulations, AlphaFold, or other sources. This way, we analyze differences in the secondary structure contents and can interpret the results easily and validate the high-resolution models. There is another approach. We also used to do it in some of our previous works, when the experimentally measured CD spectra are compared to the spectra that are calculated for the model structures by a suitable algorithm. In this case, it is hard to interpret the spectral differences between the experimental and the generated CD spectra. On the left top, there is a statistics on the performance of spectrum calculating methods using the RMSD between the calculated and the reference CD spectra for disordered and globular proteins. This was published by Nagy et al. How can we interpret the accuracy of such approach? To answer this, interestingly, the authors themselves used the other direction. They analyzed their calculated CD spectra with BeStSel and with their own ways to tell how the spectral differences are manifested at the structural level. This is shown at the bottom. Yes, the final goal is always to compare the structures. Thus, we suggest the simple approach and do the comparison directly at the structural level. On the right side, there's an example. The experimental spectrum is the blue one, and we show two CD spectra for model structures that have the same RMSD to the experimental spectrum. This RMSD value is exactly what is the best RMSD on the left side. Which calculated spectrum is closer to the experimental one? Hard to tell from the spectra. A simple RMSD doesn't tell much. Moreover, it matters what part of the spectra differ. However, if we do the comparison on the structural level by analyzing the experimental spectrum with BeStSel and compare the results to the model structures, we clearly can decide which structure is validated by the experimental measurement. The secondary structure calculation for PDB structures module of the BeStSel web server can facilitate the structural comparison, which is now available for all structures in the PDB. Hopefully, within a few weeks, this module will operate on any PDB and MMCIF files and for AlphaFold models. Here are examples highlighting the importance of experimental verification of AlphaFold structures. AlphaFold does not account for experimental conditions, mutations, or post-translational modifications that can significantly impact protein structure. As illustrated here, the same polypeptide chain can adopt different conformations, which AlphaFold cannot predict. On the left, we see alpha-synuclein, a natively disordered protein linked to Parkinson's disease and other synucleinopathies. AlphaFold erroneously predicts a high alpha helix content with high confidence, whereas CD confirms that the protein is actually disordered under native conditions. It can form beta sheet-rich oligomers, and under extreme conditions in the presence of high trifluoroethanol concentrations, it adopts an alpha helical structure, though to a lesser extent than AlphaFold predicted. On the right, we can see amyloid beta peptide, which is disordered in its monomeric state, but can adopt various aggregated structures. While AlphaFold's prediction is accurate for the monomer, it cannot model intermolecular interactions and aggregation reliably, despite its presumed direction of development of AlphaFold. Another example is beta-2 microglobulin, the light chain of MHC1, which is associated with dialysis-related amyloidosis. A point mutation in this protein causes hereditary systemic amyloidosis. The X-ray structures of the wild-type and mutant proteins are nearly identical, yet their stability and amyloid formation tendencies differ significantly. Here, CD spectroscopy and secondary structure analysis show that the mutant is more sensitive to pH changes. The recorded CD spectra as a function of pH reveal structural changes that could not be studied computationally without experimental validation. A remarkable example is insulin, which undergoes dramatic conformational changes depending on the environmental conditions. Its structure can vary from highly alpha-helical native state to a disordered structure and different beta sheet-rich aggregates and amyloid fibrils. Two selected structures are shown here. This is yet another example demonstrating the need for experimental verification of bioinformatics predictions. The disorder predictor IUPRED suggested high disorder for two regions of MLL4, a histone lysine methyltransferase. At the top panels, the blue curve above a value of 0.5 indicates disorder. However, CD spectroscopy confirmed that these proteins have ordered structures with significant alpha helical content. The molecule on the left even exhibits cooperative thermal unfolding as evidenced by CD thermal scans. CD spectroscopy is also valuable for studying protein-protein interactions. Here we examine S100A6, a calcium-binding protein involved in cancer metastasis. It binds strongly to various partners, and its binding affinity was assessed using isothermal titration calorimetry and fluorescence polarization. CD spectroscopy revealed that the initially disordered binding peptide adopts an alpha helical conformation upon complex formation. After these examples, let's see the status of the measurement in the lab.
Hello again. The measurements are completed, and now we have to go further with the data processing. First, we subtract the baseline from the sample spectrum, and then we take the data to another computer because there's no internet on the computer of the instrument, and then we will do the BeStSel analysis.
Before subtraction, we quickly check the photomultiplier voltage. The HT is below the limit in the entire wavelength range for both the sample and baseline measurement, which is good. And we also look at the region between 250 and 60 nanometers. The two spectra are overlapping and close to zero. After subtraction, we will get a nearly zero value here. So we can do the subtraction. Here it is. We save it and we can transfer it to the computer for BeStSel analysis. Waiting for the data to arrive, let's get back to where we started. CD spectroscopy is a fast and cost-effective tool for analyzing protein secondary structures under diverse conditions. When combined with BeStSel, it can validate high-resolution structures and structural models for different protein variants and conditions. This is particularly crucial for bioinformatics tools like AlphaFold. AlphaFold is a groundbreaking method, but to maintain its high-throughput capability, rapid experimental validation is necessary. CD spectroscopy with an accurate analysis tool such as BeStSel offers the fastest experimental verification, delivering results in just 30 minutes. I'm sorry, it should be the data.
Here's your data. Thank you.
Here's the BeStSel web server with all its modules. Let's analyze quickly the baseline subtracted spectrum using the single spectrum analysis function. The spectrum is saved in text format, which BeStSel can read. We choose the measured ellipticity and type in the concentration, the number of residues, and the path length. The most difficult part is to type the CAPTCHA, which we hate, but it is necessary. And then we can submit with one click the data, and we get a data examination page first, where we can check that the data is correctly uploaded. It is already normalized in delta epsilon units. Then, with a single click, we can get the secondary structure estimation. I hope we could demonstrate that it is possible to collect good quality CD spectra and analyze it for the secondary structure composition by BeStSel in half an hour. If you are interested in using BeStSel offline and you have the box, JASCO provides it with their new Spectra Manager software. If you're interested in how to do CD measurements, please refer to our method paper. About the BeStSel method and web server, please read these publications and the tutorial on the web server. How to measure protein aggregates and amyloid fibrils by CD, please see our new protocol paper. For disordered and ordered protein classification, you can read our Frontiers paper. András Micsonai is not a VIP, but the MIP, the most important person in this project. He himself did all the programming and built the website. We thank all the present and former group members for their work and company. We thank all the Hungarian and international collaborators for the long years' joint efforts and friendship. We are also grateful to JASCO people for our collaboration. This image is a never-appeared PNAS front cover design. Thank you for your attention and interest in BeStSel, and please feel free to ask us if you have any questions or suggestions.
First, we are excited to introduce our limited time CD trade-in program. Trade up if you currently own an older J700 or 800 series instrument. You have the opportunity to upgrade to the state-of-the-art J1000 series. In addition, we will accept competitor instruments as well. This promotion is available exclusively in the United States and Canada. For further details, please reach out to your local sales representative. Additionally, we are pleased to announce the upcoming establishment of our inaugural CD Users Group meeting. This event will be hosted at various locations across the United States. We encourage you to stay tuned for a meeting announcement in your region. Now, we will proceed with the Q&A session. If you missed it in the introduction, you can ask questions by clicking the Q&A icon at the top of your screen. Okay, so first question, is there any way to process data with BeStSel offline?
So at the moment, we have a license agreement with JASCO. So if you want to use BeStSel offline, your only chance is to purchase the software from JASCO, the Spectra Manager software, which utilizes the BeStSel offline.
Okay. Can CD be used to examine virus particles that have capsid proteins?
I guess it's possible if the virus particles are, we might say, single particles and doesn't scatter too much the light, then doesn't form large aggregates. So then if you have a clear solution, it could be used. The only problem might be the presence of DNA in the, or nucleic acid in the virus particle that might strongly affect the signal. So it depends on the fraction of nucleic acid in the sample. Or maybe if you get rid of the nucleic acid from the virus shell, the protein shell, then definitely you can analyze it. Yes.
Okay, next question. How to verify estimated secondary structures are correct? Is there any quality parameter other than NRMSD? Is NRSMD alone good enough to ensure correctness of a secondary structure estimation results?
So we are developing a parameter. Hopefully it will come out soon. So we continuously develop the BeStSel, and we are working on it. So the NRMSD and RMSD are not the best parameters to tell the accuracy. So what we can rely on is the statistics on the reference data set with a cross-validated way, where we can see the accuracy. So the NRMSD and RMSD are not directly related to the error of the secondary structure estimation, and it's the same for other algorithms, and maybe some algorithms are much worse than BeStSel. For this, like the CDS-STR algorithm goes for the perfect match upon fitting. So, but in structure, it can give you a huge difference from the correct answer. So for that algorithm, people often like that because the fitting is always perfect, but there the RMSD and RMSD has nothing to do with the accuracy. So hopefully in the near future we can give you a better score for the accuracy, but now you can rely on the statistics on the reference database. Thank you.
Okay. How reliable are the results obtained for shorter peptides, 10 to 30 amino acids?
So if the peptides have amino acid composition which is similar to globular proteins, then we guess it's pretty usable. But sometimes peptides have very unique composition, amino acid composition, then some unusual contribution might give a large part of the signal. In that case, the accuracy of structure estimation is not that good. And for the peptides, I also have to emphasize that we always see the average spectrum, and we can estimate the average structure by CD. So if someone uses, I don't know, molecular dynamics simulations and can see their formation of alpha helix, but it occurs only in the fraction of the time, then we will not see it by CD. So this also has to be taken into account.
Okay, so for the analysis of an unknown sample, is there a way to know the number of peptide bonds?
So it's a question if we know the sequence or not. So, okay, probably we don't know the sequence. So in that case, we can use we can determine the concentration in some independent ways, and we might estimate the concentration of the peptide bonds in our solution, but we cannot tell the length of the chain from this. So the residue number of one molecule we cannot tell. We can tell the overall concentration or the overall peptide bond concentration, but not the length of the chain.
Okay, we have time for one or two more. So how well can CD capture changes in MABs that have been exposed to thermal stress?
Well, from the change of the structural composition, you might have an idea of the structural changes, and if it's for instance an increase in the parallel beta sheet content, that might indicate aggregation, and there are different indications for that too in the signal. So if you can see some light scattering or maybe shrinking the signal, then that's definitely aggregation upon the thermal stress. If there's no aggregation, but there is a conformational change, which probably rare, then you can compare the structural composition before and after the treatment, I guess.
Okay, and last question. Can BeStSel be used to study oligoria foldamers? Sorry, I'm not familiar with that first word. OLIGOUREA foldamers.
I guess not. I'm not so familiar with foldamers, but they have a different chemical composition. So I don't think you can easily use. If you use some foldamers with peptide parts and you change them or replace them, you might
check the difference, measure the difference between them, and if it's related to change in the protein part, then you might use it. But basically, BeStSel is only good for the L amino acids, so peptides composed by L amino acids. Thank you.
Okay, so that will conclude our webinar. Thank you again, Dr. Cardos, for joining us, and thank you all for attending. If you have follow-up questions, please feel free to contact your local JASCO representative, and they will be able to get answers for you. Thanks, and have a great day or night, depending on where you're located.
Thank you very much for the opportunity, and you can also send us emails about questions regarding the BeStSel web server or the use of BeStSel. And yeah, and we are waiting for that. Thank you very much.